Welcome to this lecture of doing sequence alignment using the global and local alignment algorithms of Needleman-Wunsch and Smith-Waterman. This part of the lecture is mostly based on the bioinformatics algorithm to do sequence similarity and sequence analysis. So most of the algorithms you will get in the very good book by David Mount. There was sequence and genome analysis, and it's in Amazon. And first of all, to start with the pairwise alignments, the basic question about genes or proteins is like to what extent a pair of sequences are similar. To find out these are kind of aligned in a certain manner, after which a similarity score can be computed. In order to understand the sequence alignment, it is fundamental to have some idea of recursion. That's why the first step I just showed how to do the recursion. So initialized a vector, numeric x vector, then I started looping from the second position, since if you start with 1, so 1 minus 1 is 0, so there is nothing 0, there is nothing in the 0th index, so it will give an error. So for that you have to start the loop from the second position and then loop it down, and it uses the previous value. uh, it uses the previous value here, like 2 into xi minus 1, like 2 minus 1, the first index value, and computes the result and puts in xy. This is how the recursion works. So the recursion is one of the major uh, uh, kind of what I will say is a algorithm where you need to use it in doing this dynamic programming. Uh, for this, first of all, I will be doing the global alignment, which is in Needleman-Wunsch algorithm. And to do, like, I have like for two sequences. For example, like GAATTC and GATTA, the DNA sequences, and and there's a match between the two letters. If there's a match, so we would give a score of +2. If there's a mismatch, I will give a score of -1. And then if there is a gap, for example, aligning a sequence using globally, you need to include gaps in your sequence. For that, you need to have a penalty for that, that's around d equals 2. For example, I have taken d equals 2 here. So first of all, I have used the two sequences, and I used S2C to convert it to characters, S2C to convert into characters. Then I uh, made a matrix of these two lengths, as you can see, and then S. So let's see X. What's X here? X is converts S sequence this single type characters, and so as Y, and S is this. I made a matrix of NA values of 5 cross 6, 5 rows and 6 columns, and then I use this one. So for example, if there is a match between the two here, if there is a match between two, I give a score of 2, else I will give a score of -1. So this will initiate the first matrix. Mm, uh, so when I see S here, sorry, so this is the initial matrix, and um, next we will use the gap penalty and the max function uh, to compute the maximal score of these two aligned sequences. So again, if you don't understand this algorithm, you should go to wiki or that book to see, or search in Google how the algorithm works. And uh, First of all, I will initialize a matrix here, and then D is the gap penalty, and then this is the F1. So if the rows, the first rows, so initializes all these things, and the first as this values, and then the max will will give you the result of those which, like, see if this thing, so it takes these three values and returns only that value to Fij which has the maximum value. So after this computation, we just find this the matrix, the scoring matrix is generated, and the optimal value score between the two sequences is this last value. So that's 5. So this is how, like, you can do. I've coded this part using the using normal these functions to get this optimal score. And again, now I'll be using this Biostrings package, the pairwise alignment function, to compute the score. So it doesn't generate these kinds of matrices. So it generates only the alignment along with the score. So let's see. So S is the substitution matrix which I have used here. So here just something like this. So match is 2, mismatch is -1, and since base only is true, it means that I am assigning ATGCs, not protein sequences. So, and also the gap extension penalty is -2. So you got to see what's gap opening. Like when you put a gap, so that's gap opening, and when you extend the gap, that's -2. So you can set different values to the gap opening and the gap extension penalty. It's up to you. So after aligning this global alignment, so it score comes around 5. So it means that if we set this values here, will give a similar score, like here what I have got, and what the pairwise alignment function we got is the same. So this is how you can do the sequence alignment, and you can run this code on large sequences, like download 2 sequences and do pairwise sequence alignments of each sequences, and then you can put like create large sequence similarity, gene sequence similarity matrices, and you can use the similarity values to to do a lot of clusterings, and you can do a lot of things. So these are the much more extended algorithms, or which you can do. Like once you have a similarity score, then you can normalize the matrix. Then you can develop like principal component analysis to find which genes are, which genes are kind of similar or grouped together, and then you can do hierarchical clustering, you can do k-means clustering, you can do a lot of other things, and you can group like sequences from different species. For example, Synarbitus elegans, human sequence, mice, and the human sequence, and do find which are sequences kind of similar. So these are the further extensions of the sequence similarity matrices. What you can do. Now coming to these are for the gene sequences which comprise of only ATGCs. Coming to the protein sequences, they have this the substitution matrix what we have used earlier is 2 1 2 1. So for this protein matrices, so there are 20 amino acids, and for that they have different substitution matrices. One is the PAM matrices, another one is the Blossom matrices. So the Blossom 62 is the most kind of general matrix, substitution matrix to do protein sequence comparison, and I have used it from the NCBI what they have provided, and you can see the Blossom matrix here. So this is whole matrix with all the amino acids, and these are single letter amino acid codes, and the substitution values here. So it's the same thing. The matrix will use the substitution values and compute. So for example, if there is a and a, so take the value as 4. If there is a along with r, take the value as -1. So if there is a with n, take the value as -2. So it uses this coding matrix to compute the similarity of two protein sequences. So as usual, I have used this substitution matrix here, and these are the two, for example, two protein sequences X and Y, and this is S. So I substituted, I took out, substituted, used this matrix, and I extracted only the letters which I want using Y and X, and this is the my subsetted substitution matrix. I put a gap penalty of like T, and make a final matrix, which is the full. Let me show you. It's full of any values. I initialize it, then and when you look at F again, so initialize the matrices with some of the values here. It's a 0, -8, 16, 24, all these values here, and then I give the row names as similar. Then I run the normal algorithm, and it gives me the final score of -9 here. That's the sequence of the similarity of two sequences, and the optimal score is -9. So same as for using the pairwise pairwise alignment function, I use this, and the substitution matrix is +62. This is the score. The score is similar. So my function and this pairwise alignment is kind of have similar values here. Now coming to another most important thing is what we have here is computing the probability. Is like for example, so in doing the gene sequence alignment in the previous example, we have achieved a score of 5. So uh, uh, so is this score is optimal or not, and it's large or not? So a manner to answer this question is by comparing with alignment score of random sequences. That is why we compute probability of alignment scores larger than that score, like greater than 5 or greater than -9. So to illustrate like to how to compute the probability of scores larger than -9, we sample randomly from the names of the amino acid. The names is like P, A, W, and H, A. We randomly sample. For example, here I have sampled this 7 and the 10. So for example, and for y equals 10, and then x is 7 and y equals 10. Since it's like 10 amino acids, and this one has 7 amino acids, and compute the maximal alignment score. And this one is repeated like 1,000 times, and the probability of optimal score greater than 1 is estimated by the proportion. Like so we generate random sequences of 7 and 10, and compute the sequence scores, and then put it into a vector. Mm, then sum all the probability score, uh, sum the probability score if it's greater than score by 1,000. So it gives you uh, the probability values. So once we do this, it will take a little bit of time. Remember to use the score only, which will allow you to write only the score values what we obtain as the optimal alignment scores in the this probability score vector. And then once it's done, so all probability vector has that value, and then you sum it and just figure and find out uh, uh, if and then sum score by 1000, it will give you the probability values. Okay, it's done, and let's see, it's 011. So it indicates the probability scores larger than -1 equals 0.01, and is therefore okay, kind of small. So it means maybe the alignment is it's not so strong. If it's like it's less than 0.05, we might tell it's very strong. So we might take this 0.05 as the basic end point for like hypothesis testing. We take 0.095% confidence levels. So for this is it's greater than zero point zero 0.05, so it indicates it's not so strong. And to make it much stronger, uh, uh, what you need to see whether it is like below zero point zero 0.05 or not. So this is how you do the sequence alignment, and to find whether that sequence alignment score is greater, it's larger than some, you know, than larger than the optimal score. So the score you obtained is kind of optimal or not. So this is another version of the local alignment, which is the normal. It's not this version. So it's the full version. How you do in R. So I take two sequences. I use the Blossom matrix, and the gap open penalties is 8. I assign the matrix. F1 is all 0. Then try to see F here. So first of all, this one is initialized with an extra column of zeros here. Check it, and then so this is a different sequences. So yeah. So this is you can subset it, and this is the final score of the matrix. And for this too, you have this since it's local alignment, it doesn't take the all sequences in a provided alignment. It actually locally aligns the part of the sequences. That's why it shows you this value. This is that. So it means you have to type equals to local to do local alignment. The function is same, but change the type to local. So for the same thing, you have a score, uh, and probability of computing the score. So it's 1. So it indicates that this is not much significant enough. Yeah. You will get the codes in my in the GitHub account. In the I'll show you. It's here. DHDHD folder and our the alignments